start the discussion on this introduction to computer-aided drug design. Basically, we have covered one of the top subtopic of this computer-assisted drug design is uh, QSAR. So QSAR is basically quantitative structure activity relationship, which is generally kind of ligand-based drug design. So in this tool, we are generally discussing. We have discussed that if we have certain kinds of compounds with us, then depending upon their descriptors, such as physicochemical descriptor, lipophilic descriptors, and some other kinds of descriptors, we can come to know that what can be the kind of chemical equation which can helpful for us for the prediction of the activity of certain compounds. So this is basically for a kind of something computer assisted technique, because it is also called as CADD, and hence it is involving the use of computers. Now, the computers which are used for drug discovery approach, these are generally different from the kind of the computers that we are using. Although all the kinds of computers are somehow based on silicon chips. So these techniques are also called as in silico techniques, because they are involving the use of the computers, and these computers are made up of somehow kind of silicon chips. Okay, so that's why they have been termed as in silico drug design. That we know that whatever the biological investigations that we are carry out in our pharmacology practicals, so these are generally termed as something what is called in vitro kind of or in vivo kind of analysis. The practicals you are performing in microbiology, so those are called as in vitro, because they are carried out outside of the body conditions, right? And another kind of experiment that is so what called as in vivo. So in vivo is a kind of experimentation technique that is generally we are carried out on either rats, animals, mice, etc. So that's why it is termed as in vivo. And this technique that we will be learning, these are involving the use of the the computers, and that's why they are termed as in silico. Okay, so there are several types of the techniques which are useful where computers are taken to help of the drug design. Now, what are the advantages of utilizing computers in the field of drug discovery, in the field of drug design? So what is the meaning of generally what is so called this drug design? So friends, they say if we have fashion design, so what is we used to do in fashion design? In fashion designs, we generally used to perform a kind of analysis, kind of there's a fashion competition hota hai. So people we are used to wear different clothes. They try to customize their clothing either with the hats or and so on, and they try to give some different, some innovative look to their own. So that way, this fashion show is there, and that is called fashion design or designing of the clothes, etc. So similarly, we can have our drug design as well. So basically, it is a kind of design of a drug. The drug molecule should be in such a way that it should have certain efficient biological activity and minimum side effects. Okay, so that's why this drug design is there. And there are two terms which are associated with drug design. What is so called as rational drug design, and other is called as traditional drug design. Okay, so traditional drug design, what? That's a traditional drug design. So traditional clothes, so whatsoever the clothes is which we which our forefathers used to wear, such as turban, hats, and so on, those kinds of clothes are called as traditional clothes. So similarly, traditional drug design means the kind of drug designs which is known since the beginning of the drug design. जब drug designing field का existence हुआ था, जब drug design field मतलब इट वॉज देयर, सो उस टाइम से लेकर जो जिस टाइप का जो ट्रेडिशन चल रहा था, दैट काइंड ऑफ ड्रग डिजाइन इज कॉल्ड एज ट्रेडिशनल ड्रग डिजाइन. But nowadays, this traditional drug design is not that much popular. Why? Because traditional drug design involves so many traditional methods, and these traditional methods are much costly. And we do, and especially for our country where India is there, which is one of the developing country, so we cannot afford so much amount of money as a part of the cost behind drug discovery paradigm or drug discovery program. So that's why it is necessary that we should reduce the cost, right? And to reduce the cost, this computer aided drug design is one of the best technique. Likewise, if we are in search of certain molecule and we want to take the help of computer aided drug design, then we can use these techniques to with and can achieve this drug discovery in a low cost. So this is one of the advantage of this kind of program, what is so called computer aided drug design. It is abbreviated as CADD. Sometimes some people also pronounce it as CAD. So there is nothing wrong whether you say CADD or you say CAD. So both are similar to each other. Now, what is the principle behind this kind of CAD? So this principle is generally, which is on your screen, that we are interested in finding such a right key for the lock. Likewise, we have so many locks, but if you want to open that lock, we will require only certain key, right? At our home, there will be so many locks to each and every room, probably, and there will be a major lock at our main gate of our house or home, right? So there will be only one key which can open that lock. Definitely, we will have some spare keys of the same in our repository as well. So, but the point is to tell that if you want to open a certain lock, there is a specific key behind that. So this drug design is also for that kind of approach. What is so called to find the right key for the lock. Now, what is our lock in drug discovery? The varieties of proteinous targets, including enzymes, receptors, ion channels, and transporters. All of them are either polypeptides or peptides, and nucleic acids as well, such as DNA. So these are also our targets because in DNA is targets, or that is one of the nucleic acids are the targets for design of anti-cancer agents. Okay, so these are the various targets which are proteinous. So those are our locks. And if we want to open, if we want to interact, if we want to handle the enzymes, there will be a certain key, and that key may be our agonist or antagonist, which may be our enzyme inhibitors or promoters, or it can be chain blockers. The point is to tell that these proteinous targets such as enzymes, ion channels, and all these receptors, DNA, so those are considered as somehow kind of our drug targets. And these drug targets, which can be worked out, which can be regulated with the help of certain uh, keys, and these keys are nothing but they are generally termed as our these kinds of uh, inhibitors, agonist, antagonist. It can be blocker also, depending upon what kind of channel is there. If it is sodium channel, then we can call it as sodium channel blocker, potassium channel blocker, and so on. It may be also calcium channel blocker as well. It can be certain a kind of uptake inhibitor. We know that several kinds of uptake inhibitors are known for varieties of neurotransmitters such as noradrenaline, adrenaline, or some others. Maybe selective serotonin reuptake inhibitors. So SSRIs, those what are terms as uh, there. So the point is to tell that these are the various kinds of the locks which can be handled with the help of their uh, structure. So जैसे if we want to open a certain lock and key is missing. So if you want to open up the lock, that lock, so there are two ways. Either we will break that lock or else we will make a duplicate key of the same. Okay. So this, how can we make a duplicate key? So just say if you want for a simple lock of at our home whose key is lost, we want to make a duplicate key. So we will go to the market. We will catch some person who is expert in making the duplicate key from the locks. So then we will approach him and we'll ask him to do this thing. So at our home, he will make a certain rod, and that using that rod he will fit into that key and will try to in the lock, and he will try to do give the shape to that rod, so that rod can be converted into a duplicate key. So same way drug discovery is also that we will do some kind of techniques that a simple molecule which is with us, so we will try to see whether a molecule is fitting into that lock, into that protein or not, and then we will try to give the shape so that we can work with the desired our protein target. Okay, so this is the major goal behind the structure-based drug design. That if we have a proteinous structure, then if we come to know that what is the active binding site and what is the molecule that is going to interact with that protein, so we can find out a new molecule which can change the protein's activity. Either if it is agonist, then it will increase the activity of a protein. If it is antagonist, then it will decrease the activity of the receptor. So there are several receptors are known, such as G protein coupled receptor, or it may be ionotropic receptors, or it can be kinase link receptors, or it can be nuclear receptors. So these are the several types of the receptors which are known and can be considered as a proteinous target. Okay, so this is one of the major aim behind the structure-based drug design, structure-based uh, drug discovery approach. So our purpose is we want to do lead optimization. If we have so many molecules, thousands of molecules with each other, and we want to narrow down these thousands of molecules into best 10 molecules, then under these conditions, this drug discovery approach can be utilized. And it involves so many techniques such as QSR, quantitative structure activity relationship, which we learned in our some of the previous classes. Molecular docking, so that is also a kind of the approach we will be looking at to this, and somehow inside we will be having during our practical session as well. Molecular dynamics, so that is a kind of technique which is a sub part of molecular docking. So using certain software, some of the softwares which are freely available on internet, and some of the softwares which are paid version, so we can work it out with them to develop the molecular dynamics of particular proteins. Pharmacoform mapping, so that is also kind of technique which is used in structure based drug design. Now, what is this pharmacoform mapping? Just like we have our Google Maps, right? So our Google Map is what? If, for example, if from LM College of Pharmacy, if I want to reach to, let us say, Delhi Institute of Pharmaceutical Science and Research, DIPSAR at New Delhi. So there will be a specific way to go to that college from our LMCP. So if there is either if we can go by the foot, we can go by road, we can go by air, or these are the ways that, or by rail also, or by train as well, right? So these are the ways that using that we can reach to our this destination. Fine. So now the question is that how to go there. So Google Map can help us if we want to buy our personal driving car. If we want to go from LMCP to Dipsar, then we can go using Google Map. Just say we can drive the car in the suggested routes, in the suggested map routes using Google Map. So what is that? So that way, it is a kind of mapping that it will show certain roads that yes, this way we should turn, this way we should go ahead. After certain kilometer, there will be a certain fly or flyover bridge, or after certain time there will be a toll will come. booth point will toll booths will come. So that these are the different kinds of mapping of our destiny towards our destination for the specific routes. So similarly, this Google, pharmacoform mapping, that is a kind of mapping technique for pharmacophore. So what is pharmacophore? Pharmacophore in general, it is mean as certain kind of key features which are present in the molecule to interact with the desired target. So just like alpha and beta adrenergic receptors, there should be certain catechol moiety present in the structure. A phenol ring or a benzene ring having 1,2 dihydroxy substitutions. So that kind of feature is one of the pharmacophore feature to show the interactions with adrenergic receptors. So adrenaline, noradrenaline, all these neurotransmitters possess catechol moiety in their structure, and they can selectively interact with alpha and beta receptors. Okay, so this catechol moiety is termed as a pharmacophore. For means a kind of functional group and which can show certain pharmacological activity, which can show certain biological activity. That is so what called as pharmacophore. So we want to map, just like Google Map, we want to map that what can be the particular pharmacophore which can show the interactions. Let's say from LMCP to DIPSAR, what is the distance? Let me tell you, 14,000 kilometer, 1400 kilometer, 1,400 kilometer, 1,400 kilometer. So if that is a distance between LMCP and DIPSAR, situated one of the institutes situated in Delhi, New Delhi. So the question is that what can be the distance between? So if that is a case for Google Map, so the for pharmacophore that we can map that what can be the distance between the 2 points in angstrom, and what can be the distance between the residue 1 and other point, so that we can map. And then the third point and the first point, what can be the distance? So all these kinds of the distance we can map, and this is possible because of the advancements of so many kinds of spectroscopic techniques which can allow us to predict the structure of the compound, the structure of the protein. Let's say Dr. Podiwala might have taught you several classes on this identification of or certain spectroscopic techniques such as nuclear magnetic resonance. That is one of the spectroscopy that can help us to predict the structure of the compound, what is called as NMR. So this highly sophisticated technique that is also can be used for prediction of the structure of large molecule, such which is peptide, polypeptides having molecular weights in kilodaltons. So that we can use. We can also take the help of XRD, X-ray diffraction spectroscopy, which is one of the spectroscopy involving the use of X-ray radiations. We are very much aware about as a pharmacist what are called X-ray radiation, one of the radiations which are very highly energetic and powerful radiation, because these are the radiations which are useful to know the defects in the bones that some person who met the injury, and we want to identify which part of the body has undergone fracture, so we can do the X-ray X-ray can take the X-ray of particular organ. We can subject that organ to X-rays. So X-rays will give us idea about the texture or the pattern of the fractured bones, right? And then by looking at X-ray spectrum, we can identify that so and so portion of the X-ray or so and so at this portion of the organ is defective, right? So this is the use of X-ray radiations. So if X-ray radiations can cross the bones, which are much enough stronger, made up of calciums and phosphates, so these X-ray radiations can also be used to analyze the structure of uh, some these molecules which are called as peptides or the proteins. Okay, so with the help of these techniques, this structure-based drug design is developed, and we can derive the first. Once we have the structure of the protein, then very well we can deal with them. So this is one of the goal of structure-based drug design. Our purpose is we want to increase the activity of the molecule. We want to search a new molecule, just like COVID-19, coronavirus disease 2019. That is one of the bigger, I will say, a kind of bigger enemy of the human society. So if we want to kill, if we want to fight with this enemy using a certain potent medicine, then this such techniques such as molecular docking, pharmacophore dynamics, pharmacoform mapping, and 3D QSRs can be used to find out a new molecules which is going to act against COVID-19. So keeping this in mind, Government of India has recently released a somehow program what is called as Drug Discovery Hackathon. So this Drug Discovery Hackathon 2020 is one of the kind of such program where we students like you and the faculties like us can take part as a part of somehow kind of this uh, drug discovery against COVID-19. So that is one of the approach that you PharmD third year students are also eligible to take part in this drug discovery uh, hackathon as well. So if you know what is called structure-based drug design, then you can very well take part in such national level competition. Okay, so let us learn this what is called structure-based drug design. So structure-based drug design, that is termed as SBDD. So this SBDD can be divided into these two parts: target-based drug design and another what is called ligand-based drug. What is called target-based drug design? So target-based drug design is a kind of approach that if we know the structure of a protein, then this structure, once we identify and it is having certain kind of active site, so the molecule which is going to bind with this such a protein and can show the inhibition. So this one of the first success has been identified by Robert in the year 1919, and using this target based drug design of the protease enzyme, he could develop this HIV, human immunodeficiency virus protease inhibitors. So, and it was a peptide drug. It was made up of somehow amino acids. So that he could identify not more than I will say 30 years ago, right? So 30 years ago, using this target-based drug design, Robert has identified this HIV protease inhibitor. So how this HIV, what can be this target-based drug design? That if we know the structure of the target of a protein, then we can design our this peptide, and that peptide will make peptide drug inhibitor complex. This is a receptor, this is a ligand, so it will make certain complex. So that complex can be drug receptor complex. Okay, so this is one of the technique where we know the structure of our target. Target means our protein. So if we are discussing of COVID-19, so there are so many kinds of proteins are known, such as ACE2 inhibitors, angiotensin converting enzyme 2 inhibitor, M-pro protein, spike protein, the RNA dependent RNA polymerase protein, and several kinds of other proteins which are reported for which can be one of the integral composition of COVID-19, right? So that way, this proteins, if their structures are known using XRD, using NMR spectroscopy, then we can design such compound which can inhibit the biological processes which are going to occur in COVID-19, and then we can inhibit the formation, we can inhibit the transmission, we can inhibit the survival of COVID-19. I mean to say virus, and then we can win against this enemy. Okay. So another approach after target-based drug design, that is called ligand-based drug design. So it is ligand is what? Ligand is basically the inhibitor. Ligand is basically the drug molecule. Likewise, we note that remdesivir is one of the molecule which is acting against COVID-19. That is used as a drug. It is injected to the COVID-19 patients into their body, and to which is going to inhibit the protease enzyme. Okay, if we know that, then we can create the somehow analogs of remdesivir. जैसे कि in this structure of thymidine there was hydroxyl, so we replace this hydroxyl with this azide N3, and this is basically a zidovudine which is going to act against HIV virus, human immunodeficiency virus, and the activity of zidovudine is far better than thymidine. So this is a kind of approach that is called ligand-based drug design, because here we are not claiming the target of the protein. Here we are not discussing the mechanism of action of this compound, but or mode of action of this zidovudine, but just by replacement of hydroxyl group with azide functional groups, we are going to derive a potent molecule. So given a structure of a ligand, a new molecule can be predicted to change, to alternate the protein's activity. So that is so what called as ligand-based drug design. There is difference in target-based drug design. In target, we are dealing with the structure of the protein, the structure of the virus enzyme or the receptor, and ligand-based drug design where we are dealing with the structure of the inhibitor. We are dealing with the structure of the ligand, or not only one ligand, it can be more than one ligand. So this approach is called as ligand-based drug design. For COVID-19, if we want to apply this approach, then we can take the structure of remdesivir and can design the potent derivatives of remdesivir. We can take the structure of chloroquine, hydroxychloroquine, and then we can further design more potent compounds for COVID-19, and which will be more potent than this chloroquine or hydroxychloroquine. Fine. So these are the approach where we can take the help of computer aided drug design. So just by using our computer, mouse and keyboard, so this way we can do the drug discovery. Okay, if you visit this drug discovery hackathon, then you will identify the varieties of different things which are being carried out from under this thing, and we can do something new, what is called drug discovery approach, drug discovery hackathon. So you search on Google, the link is already shared with you, and CADD is one of the technique where you can help take the help of these techniques such as whether molecular docking or pharmacoform mapping or ADMET filters for in search of certain potent compounds. Although remember these words, the field of research is not straightforward approach. You will meet with so many failures. You will have to give lot of time to that particular kind of drug discovery, and in the end you will have a single molecule which may be active or which may not be active. If it is active, that it can be carried forward for the purpose of synthesis, for the purpose of biological testing or evaluation, and clinical investigation can also be carried out. If it is not active, then at least you will have some idea that yes, this is not the working way. There shall be some different way than this. Okay, so computer aided drug design is one of this approach where if we know the structure of the light, this look at this table. This table itself indicates where and under which conditions, which computational techniques can be useful. Okay, so for example, if you know the structure of the ligand, only one ligand or more than one ligand, then you know the structure of the protein, then in this context you can adopt structure based drug design, especially this molecular docking. Docking is a kind of technique where we can dock our compounds. We can align our compounds to a particular site and can identify whether a compound is going to interact or not. So that we can identify using this technique. Okay. Then if secondly, this ligand based drug design, if we know the structure of the ligand, but we don't know the structure of protein, and in these conditions, ligand-based drug design such as QSAR and pharmacoform mappings can be useful. Okay, so that itself indicates that ligand-based drug design, where we know the structure of the ligand, but we don't know the structure of the protein. The third technique, where we don't know the ligand, we don't know what is the structure of the ligand, and but however we know the structure of the protein, then de novo drug design can be useful. So this is one of the technique where we can use this computer aided drug design. But let me tell you, if we don't know the structure of the ligand, if we don't know the structure of the protein, then computer aided drug design is of no use. So under these conditions, CAD cannot be useful. Okay, so the point is that these are the three techniques. We either, if we know the structure of the both the structures, then polar docking can be useful. If we know only the structure of the ligand, structure of the inhibitors, structure of the one single molecule, then ligand based drug design can be used. But if we don't know either structure of our protein, that what is the basic structure, X-ray crystal structure of protein present in coronavirus, or we don't have any medicine. So under these conditions, de novo design that can be useful. De novo design, that is a different terminology, likewise the novel. De novo meaning completely new, something additional which is not reported at a time. So that is this meaning of de novo, the novel, the something, the new, or something new which can be useful in such kind of designing. So structure-based drug design, molecular docking is there, where we know the structure of the ligand, we know the structure of the inhibitor, and the structure of the protein. Ligand-based drug design is the technique where we are not interested in protein. What is the target? What is the receptor? That is of our no use. But just like we discussed the things in QSAR, quantitative structure activity relationship, we just discussed what are the descriptors, what are the properties of the molecules, what are the properties of the inhibitors. So that we discuss in QSAR, but we didn't discuss what is their mode of action, what is their mechanism of action, how they are going to interact with the protein. So this can be useful when a ligand based drug design is there. Okay. So I hope that I am able to explain you what is that the various techniques which can be used in quantitative structure, in this drug designing techniques, whether it is structure-based drug design or it is ligand-based drug design. So friends, another technique after this, that is so what called as virtual screening. So virtual screening is one of the technique where we can virtually screen our compounds. Okay, so that is what so called virtual screening. Just like now because of COVID-19, we cannot come to the colleges, but still our education is going through with the help of certain kind of virtual programs. So virtual programs like Google Meet, WebEx, or these are or many other Zoom, and so there are many certain kinds of Microsoft Teams, etc. whatsoever they are. So they are allowing us our virtual education. Although I am at home, my home, you people are at your home, but still our process of learning is continuous since our day one. So that is possible through with the help of this kinds of virtual platform. So this education is called as e-learning virtual education. It is in fact not real because I cannot see you that what you people are doing, and similarly you cannot expect me all the time on your screen. So that's why this is virtual screening, virtual learning. So similarly, we can have a screening of the molecule, that looking at the structure of the compound, we can identify whether molecule have certain characteristics in to become a drug or not. Likewise, if I cite one of the example of my professor, my teacher, Professor Prasad V. Bhartam, has taught me this, the same example which I want to give to you. Just like if from your class, there your class may be of 30 or 50 capacity, you people are 30 there, and looking at your behavior, that we can predict whether a student will become a teacher in the future or not, whether he or she will become a professor or not. So that we can predict depending upon his or her nature, his or her knowledge, or his or her teaching or presentation skills. So that we can predict. And there's some students who are really poor, who don't have such knowledge, so definitely we can also predict that no, she is not going to make a successful teacher in the field of academics, in the field of teaching. So that we can predict. Okay, so this is all looking, जैसे आप लोगों में भी डिस्कशन होता होगा कि यस, अरे वो तो बहुत बड़ा प्रोफेसर बनेगा या बनेगी. So likewise, this we can predict from your nature, from your skills, from your capability, from your potential, that we can predict. So similarly, this screening can also we can predict using virtual tools, that whether a molecule will become a drug in future or not. So that we can predict. You have already performed one of certain filters, virtual filters such as Lipinski filters. And using Lipinski filters, you could find out that whether a molecule will become a drug or not. So those various filters you have applied. What were there? Those filters, that molecular weight should be less than 500. Hydrogen bond acceptors should be less than 5. Hydrogen bond donors should be less than 10. Then number of rotational bonds should be less than 10. The C log P lipophilicity should be less than 5. So these all filters you have applied, and then you could come to know that from your structures, from your 20 molecules, only 2 molecules cannot become drug. Rest other molecules can become a drug molecule. So that kind of analysis is called virtual analysis, Lipinski filter analysis. So that you have already performed. Okay. So this kinds of can be analyzed into two ways, that ligand based methods and structure based methods. Okay, so ligand based methods where protein ligand docking is there, and structure based method where molecular docking is there, and this ligand based methods where pharmacophore based designs are there. So basic our purpose is that जैसे biological screening होता है, so we are screening thousands molecules against particular enzyme. So virtual screening, which will allow us to screen the molecules virtually, then we can and selectively synthesize one or two molecules. So this is what called as virtual screening. The last example of this thing, that is a new one, that is pharmacophore based drug design. So what is pharmacophore? So the dictionary definition of pharmacophore is that it is a set of features within a molecule which can predict the biological activity of the compound. A set of chemical structures in a molecule that is going to the biological activity of a molecule. Just like if we want to identify any molecule to be anti-malarial one, then definitely it should have quinoline scaffold or isoquinoline scaffolds into itself. Then it can show its anti-malarial activity. If we want to predict the proton pump inhibitory activity of a molecule, then it should have at least benzimidazole scaffold in its structure. So that way, benzimidazole is a pharmacophore for such kind of molecules. Catechol, a benzene ring which is attached with two hydroxyl groups at 1,2 position, that is also a kind of pharmacophore for showing the action against alpha beta adrenergic receptors. Then a certain kind of beta aryl amines, which is essential for somehow other anticholinergic activity. So all these are the features, the pharmacophore features for particular molecule to show their biological activity of our interest. So we can generate the pharmacophore just like I gave you the example of between the LMCP and DIPSAR. So here we can predict the calculate distance between the two different coordinates, two different hydrogen bond receptors, and we in angstrom, and said let us say what is the distance between this point and this point. We can calculate what is the distance between this two and this two. Then here this is the point, this point is also of our interest. What is the distance in angstrom? We can calculate what is the distance between this point as well as here the last one and this one as with this line. So this kind of distance mapping, the pharmacophore mapping, just like Google map, कितना LMCP से ये कितना दूर है और वहां से ये point कितना दूर है. So that way we can measure in kilometers. So here the same distance is mapped in angstrom. The meaning of angstrom is 10 raised to minus 10 units. Okay, then we can derive certain structure which will fit into this pharmacophore like this, and that will exactly give us idea. हम फिर हम एनालिसिस करेंगे कि इनमें से ये जो इस डिस्टेंस है that is equal to this phenolic OH and phenyl ring, then this distance is applicable for this one is perfectly suit for phenyl ring and so functional group. So this kind of analysis can give us finally a certain compound which we can synthesize and can test the biological activity. This approach is called as pharmacophore based drug design because we are dealing with a pharmacophore, we are dealing with a drug molecule. Although my purpose is that just my objective is I want to introduce you people with this several types of computational techniques that are used under the umbrella of CADD. CADD में which are the techniques can be useful. So I want to discuss those techniques with you, and then not just an overview, not the depth in depth. If I try to teach you, it will take a lot of time, at least 5-6 years. So in this way, my objective is just to introduce you people what is the meaning of these kinds of techniques. Okay, so the last slide for this today's session, that is this protein ligand docking. So what is that? We know the structure of the protein, we know where it is going to bind. Just say what is that? Just like if we have a running classroom, then you people have decided that whatever is the whomsoever will be the teacher, but I will go to seat and will sit at the last row of the last bench or last seat. So that is for your suitable position. That either you, if you are may be comfortable at the last row or the last bench, or you may be comfortable depending upon your nature. You will be always comfortable in the first row and the first bench. Okay, if I ask the student who is regularly sitting on the first seat on the first bench, and if I ask to sit and at the last bench, then he or she will feel uncomfortable. Okay, so what is this? This is basically a kind of mode of feasibility, that molecule, although there is a big hall, big classroom is there, but you will sit at your feasible position. So similarly, we have a huge protein molecule, which is consist of so many amino acids, so many structures as well, and that way we can. But the molecule is not going to all the sites wherever it is going to bind selectively at a particular site. So that site is called as binding site. That site is called as active site. Our ligand molecule, which is our drug molecule, that will bind to this active site and will make this drug receptor complex. And this drug receptor complex, if we try to zoom, then it will look like this, where molecule has found suitably has been bound suitably fit and well into the active site of the enzyme. Okay, so that is so what call as molecular docking. Okay, and what is there in molecular docking? How can we predict the binding interactions? How can we predict the binding scores, etc.? So these are the various approaches for such as molecular docking score, docking interactions, etc., etc. So they can be also used in this protein ligand docking, also called as molecular docking. Okay, so that we will learn in our coming classes. In our coming classes, we will discuss further from here onwards that what is the meaning of this molecular docking, how it can be carried out, etc., etc. So I hope that whatever till now I am able to make you clear about this. And so in coming class, we will discuss further from here onwards. So thank you everyone. Thank you so much.